We'll wrap up our work here in Chapter 1 with this lecture on the fields of microbiology and a little bit about the history of each of those fields. So here we go. So one of the first fields to discuss is that of immunology. Immunology is the study of the the body's defense against agents of microbial disease. And we will unpack a lot of this in a chapter coming fairly late in the course. So, but it's just the idea that the body can learn what can learn the invaders, and very specifically go out and take down those invaders. So some of the history of immunology and microbiology involves a woman named Lady Mary Wortley Montagu. She was British, but had picked up during time overseas a process referred to as variolation, in which a little bit of the fluid from a smallpox blister. So smallpox is a viral disease that that can be deadly, but more generally leaves survivors with tons of scars all over their body. So if you were to go and Google some pictures of people with smallpox, it's a very disfiguring disease. And she picked up a process where you would soak a thread in the fluid from a smallpox blister of someone who was infected with this, and then make a small slice in a person's skin, and then drag that wet fluid-laden thread through that cut. And the person would then develop immunity against smallpox. Edward Jenner, who was a British physician, noticed around the same time that women, the milking girls who did all the milking of the cows, and cows have a very similar disease to smallpox called cowpox, he found that women who were the milkmaids did not have the same likelihood of coming down with smallpox. And so he began inoculating people with fluid from a cowpox blister and found that it prevented smallpox. And he is responsible for developing some of the first vaccinations using with his own son. In fact, he vaccinated his son and then infected him with smallpox and found that he did not get smallpox. Louis Pasteur, he, as we've already talked about, was involved in developing rabies vaccines and also cholera vaccines against the disease cholera, which a disease of the gastrointestinal system, led to lots and lots of diarrhea and people dying of dehydration. And then last of all, for this conversation at least, we turn to Elie Metchnikoff. Metchnikoff was a Russian working in the late 1880s, and he was the first to discover white blood cells, the phagocytes that are in the body, and capable of engulfing and destroying, consuming bacterial cells. It's an odd little story about how he discovered them. He was doing some studies of sea stars and had some live sea stars in bowls in his lab. His children came through and began torturing the sea stars, poking them with thorns, and he looked at these poor abused sea stars, and what he saw were little swarms of these phagocytic white blood cells around the injuries, the sea star injuries, and that led him to look for these in other organisms as well. And he was able to see, the work that they were doing at eating up bacteria, these phagocytic white blood cells. Okay, so that's immunology. Virology or virology, the study of viruses, is another field within microbiology. And again, we've talked about viruses. These are non-living packets of DNA or RNA that are surrounded by a coating of proteins, and they are capable of getting their genetic material into healthy cells, and then they co-opt the cell reproduction processes in order to produce more virus particles. And I have here 3 different types of viruses. There are way more than 3 types, but here are some of the, these are cartoons, of course, of what a virus can look like. So here is a bacteriophage. The DNA is maintained up here in the head of it. It can rest on a bacterial cell using, it looks almost like a little lunar lander, and then it injects the DNA through the cell membrane of the host cell. Here we see we can't see the DNA in this one because it is all completely surrounded by a protein coat. Those individual ball-looking things are individual proteins that link up and surround the DNA or RNA that's inside. And out here at the tips, we see receptors or that will bind to the cell of a host cell and then cause the host cell to draw the virus particle into the cell. So the cell kind of is helpless. It pulls the virus inside, and then the virus takes over the cell. And then last of all, here's one in which it's a long thread of virus shape where the DNA is inside, and then here are the individual proteins that make up the protein coat. All right, sorry about that. Let's go to the next slide then. Chemotherapy is another field within microbiology, and chemotherapy is the use of chemicals to combat diseases. And according to our book, roots go all the way back to the early Greeks, but what we know is that even ancient cultures, way earlier than the Greeks, were using pharmacological plants, taking plant materials and using them as medicine. One of the big fathers of the production of chemotherapy treatments is Paul Ehrlich. Ehrlich was a German, and he discovered that dyes and other chemicals could be used to kill microbial cells, but not the animal cells that were hosting the microbes. And he was able to develop a number of chemotherapies that were effective against sleeping sickness and syphilis. Syphilis was the chemotherapy is called Salvarsan. So, and I love that picture of Ehrlich in his office. So, yeah, you can tell he was a busy man. He is a big name in early chemotherapeutic work. So, a little bit more about chemotherapy. Antibiotics are antimicrobial drugs that treat or prevent bacterial infections. And some of the first antibiotics were developed in 1917 with the discovery that actinomycetes, that's a group of bacteria, are capable of stopping the growth of other bacteria. So you put them together on a petri dish, and the actinomycetes bacteria will slow and stop the growth of other bacteria on the plate. The big discovery was made by Alexander Fleming. Yeah, and some of the early actinomycetes derived antibiotics were too dangerous to use in humans, but that kicked off this idea that we could use microbes to fight other microbes. Penicillin was discovered by Alexander Fleming, or at least he recognized the potential of penicillin, which is a product that is isolated from a fungal material called, with the genus Penicillium. So for years, or for centuries again, people have used molds as a form of chemotherapy. So frequently in your home, you would keep around a piece of moldy bread, and if somebody came down with a disease, they would use that moldy bread by eating it, by putting it into something that you could eat. Would you then be able to help fight off the bacterial infections. And so Alexander Fleming, and again, another one of those happy accidents, he had some of his work get accidentally infected with some penicillium mold. And what he realized is that penicillium would halt the growth of the Staphylococcus bacteria that he had on his plates. So he discovered this in 1928, but it wasn't until World War II, and under the pressure of the need for antibiotics, that penicillin became mass-produced. Let's see. Also, and the reason that penicillin wasn't immediately, or the effects of penicillium on bacterial growth wasn't immediately pounced upon, is because sulfa drugs became available about the same time that penicillium compounds were recognized as having potential. Sulfa drugs, these are drugs that incorporate sulfur in the chemistry of the sulfa components. There was, very great development through the 30s in the production of sulfa drugs, but there was a big scandal as a number of people were poisoned, and it was because of non-active ingredients that were used in sulfa drug preparation. But that led to the Food, Drug, and Cosmetic Act of 1938 that began the regulation of chemotherapies of drugs. Let's see, some other sources of antibiotics. There are soil bacteria that yield a product called actinomycin. Actinomycin is way too toxic, but there are less toxic bacterial products similar, and one that is still used today is streptomycin. Also in the 1940s, as penicillin was being developed, neomycin, the same active ingredient that's in Neosporin, and chlorotetracycline, the first of the tetracycline antibiotic drugs, were also introduced. So we see antibiotic drugs really kick off in the 30s and then really hit their stride in the 40s. My image here is Streptomyces griseus, which is the source of the streptomycin antibiotic. Okay, so moving on now to the fields of genetics and microbiology. Working in 1928, Frederick Griffith, a British fellow, I forget, I think he was actually in Scotland, but part of Great Britain. He discovered that formerly harmless bacteria can develop the ability to become pathogenic. So can develop the ability to cause disease. And he realized and discovered that they could acquire this ability from dead bacteria. And so that kicked off great interest in studying genetics, and eventually, as molecular biological techniques came into play, the greater understanding of how genetic material in the microbial universe is moved around and acquired. So his experiments, he was working with two types of Streptococcus pneumonia. That's the bacteria, one of the bacteria that's responsible for causing pneumonia. There were rough forms of this and smooth forms. The smooth form has a capsule around it. It has this gooey material that surrounds the cell, and it can cause pneumonia. The rough form doesn't cause pneumonia. It doesn't have that capsule. It can't adhere and can't cause pneumonia. So So here's his experiment. He injected mice with live R-type bacteria, and as expected, saw no pneumonia develop in these mice. Then he would inject mice with dead S-type, the pathogenic type, and found that that would not cause pneumonia because the bacteria were dead. But when he mixed these two together, injecting into the mice dead S-type and live R-type, he found that they caused pneumonia, and recognized that it was some process that allowed living bacteria to pick up the genetic information from dead bacteria. So this is not the kind of genetics that we see in eukaryotic organisms like us. We can't go wandering through the house, picking up DNA and developing new characteristics so easily the way that bacteria can. And so, bottom line, genetic processes in microbes are unusual when we compare them to eukaryotic organisms. All right, so now we can describe then the golden age of microbiology. And if we were to try to put a timeline to this, it would be from about 1874 to about 1917. So this is the time during which, or it was the point at which ideas about spontaneous generation have been disproven, and microbiology became a really, that was the cutting edge of biology, especially because we were beginning to recognize, thanks to Koch and his work, how to isolate the causative agents of disease. And most of the bacteria that cause disease were identified during this time. And early work on viruses and viral-borne diseases was also begun. So it was the time during which we really were figuring out, this is where disease really came from. All right. So there we have it. Everything you ever wanted to know about microbiology and its scope and history. You know what to do. If you have any questions, reach right on out. Oh, dadgummit. 2 more slides. I've done it again. So that brings us then to what is it that's next? What's coming next in microbiology? Well, genetic engineering is a huge thing. And just in the past couple of years, we've developed new techniques for being able to use viruses to insert new genes into living cells. And so there's great opportunity coming to be able to treat inherited diseases and genetic conditions. And we see the use of these kinds of techniques to genetically modify food crops, especially, to either resist the diseases or to resist pest control chemicals, or even to add, proteins to, increase the protein in, say, rice or other staples, especially among poor countries, where getting access to good proteins can be difficult. So genetic engineering is one very important field that's at the forefront of microbiology. Bacteriophages, also called just phages, as a means of fighting disease as well. These are viruses that attack and kill bacteria. And so there is great opportunity available, and as we are finding that more and more of our antibiotics are becoming useless as antibiotic resistance develops among strains of bacteria, bacteriophages may be the next way that we go about controlling bacterial diseases. Bioterrorism, which is the use of pathogenic microbes to threaten or to kill large groups of people. Bacillus anthracis, especially because it forms endospores that are quite resistant, is a great subject of study and how to be able to threaten groups of people. And if you recall, sort of in the post 9/11 times, you may not recall directly, but you may have heard about it. There were several instances of people dusting mail with anthrax spores and then sending those off to politicians and other public figures. And then last of all, antibiotic-resistant strains of diseases and figuring out how to deal with these antibiotic-resistant strains. So MRSA, which is a strain of Streptococcus aureus, and that's what that image there is to the side, is one of these antibiotic resistant strains. We have very few drugs that will work anymore against MRSA, and there are other bacteria that are developing extreme antibiotic resistance. And so searching for alternative treatments and alternative antibiotics will be a big part of what microbiology is about. Okay, now we're really done. Again, if you have any questions, you know how to reach me.